Ants are one of the most diverse groups of colonial insects, with more than 12,500 species being described, and they're believed to have originated over 120 million years ago. Ever since their origin, they have become extremely abundant globally, occupying almost all continents and dominating most terrestrial ecosystems. They also account for a large percentage of animal biomass in certain environments. Ants also play very important roles in ecosystems, branching from supporting services such as soil movement and nutrient cycling to regulating services such as seed dispersal and pollination. They have relatively vast and quite intriguing interactions with their environments and other species. The ecological dynamics expressed by these organisms can be quite intricate and complex, sometimes resembling human activities. Everything from diplomacy, war and slavery, to agriculture, infrastructure, and societal hierarchies. They combat challenges as real as ours, such as amputations of infected limbs, and also face threats we would consider science fiction, such as zombie outbreaks and skinwalkers. Although ants participate in a variety of interspecific interactions with varying species, here we're going to put our focus on interactions between ants and fungi. One interaction that we will be focusing on is the parasitic interaction between carpenter ants, Camponotus leonardi, and Ophiocordyceps unilateralis, or also commonly known as zombie ant fungus. This interaction is quite complex, and being that it is a parasitic relationship, the parasite, being the fungus, will benefit at the expense of the host, which in this case is the ant. This specific fungus is relatively widespread in tropical forest ecosystems across the world, and there have also been some reports of this fungus being found in more temperate ecosystems. It is important to note that there are over 400 species of cordyceps fungi, and it does not only infect ants, but also many other insects such as spiders and beetles. However, usually manipulation of behavior is only seen in the ant species. Interactions between ants and fungus are not always harmful to one of the species involved, however. This is demonstrated by the other ant-fungi interaction we'll be covering, being the mutualistic interaction between Panamanian leafcutter ants, Acromyrmex echinacea, and fungus from the Lepiotaceae family. In this interaction, the leafcutter ants farm the fungus. Both species benefit from this interaction, with the ants getting food from harvesting the fungus, and the fungi receiving food and protection from the ants. Ant fungi culture arose in South America about 50 to 60 million years ago. There are over 200 described species of fungus-growing ants that have an obligate mutualism with the fungus they cultivate. Almost all of these species of fungus-growing ants cultivate litter-decomposing fungi in the Lepiotaceae family. The only exception is a subgroup of fungus-growing ants that farms a different wood-decomposing family of fungi, but they farm them in the same way as other fungus-growing ants.
So we're going to first start by talking about the parasitic interaction between ants and Ophiocordyceps fungus. The zombie ant fungus begins as a spore, which will then attach itself to the ant's cuticle through direct contact. Now, once contact is made with the ant, the fungus will then penetrate into the cuticle through the use of various enzymes as well as mechanical pressure, which will then allow the fungus to feed on the inside of the ant and gradually spread throughout its body. Now, after a certain threshold of infection level, the ant's behavior will begin to change dramatically. These changes include disruptions in normal behavior, such as following the colony's pheromone trails, and instead, the infected ant will walk erratically, as well as fall down repeatedly, thus separating it from its colony. Following this, the ants will experience a transition from this erratic and random walking behavior to then being compelled by the infection to climb onto a leaf vein or twig and lock onto it with its mandibles. This is commonly known as the death grip, as the ants will ultimately remain in this position and die. Then from the ant's corpse, the fungus will continue to consume the ant from the inside, then will eventually grow a stalk out of the ant's head, which will then sporulate, thus dispersing more spores onto the uninfected ants below. Now, an observational study was conducted in Thailand, which found that the location of where these ants perform the death grip is very, very precise. They found that not only did every ant perform this bite on the underside of the leaf, but also did so predominantly on the north side of the plant, about 25 centimeters off the ground. This study also found that these chosen leaves had a humidity level between 94 and 95% and temperatures between 20 and 30 degrees Celsius. This study clearly indicates that the Ophiocordyceps fungus behaviorally manipulates the carpenter ants into a niche that not only increases the fitness of the fungus, but also is very distinct from where healthy carpenter ants would usually be found.
How behavior is modified. Based on what we know regarding the behavioral manipulation of these ants by this specialized fungus, many assume that this fungus would primarily infect the head of the ant, therefore infecting its brain. However, recent studies have found out that the fungal cells were not only located in the head of the ant, but also the thorax, gaster, and legs. Scientists went further and used fluorescence confocal microscopy to look for the presence of fungal cells within the brain of the host ant. Results from this showed that fungal cells were in high abundance around the brain but were not observed directly within the brain. They then looked at the muscle cells throughout the ant's body and found that the O. unilaterus hyphae was present between and within the muscle cells in the head and leg regions and formed a network around these muscle fibers because no fungal cells were found within the brain of the host, but were found in the muscles. This may suggest that O. unilaterus does not control the minds of the ant, but instead controls their muscles. And future directions we can go with this. The exact methods on how these fungal cells actually influence muscles is still unknown and requires further research and analysis to understand the complete interaction between this fungus and its host. With current day data and knowledge, genomic sequencing of cordyceps species and their ant hosts can also be performed to provide more valuable insight into the co-evolutionary dynamics of this relationship and how these fungi have adapted to their hosts over time. Another area for improvement is there are over 400 species of cordyceps that have been identified to date, and there are likely many more, and each species has evolved to infect a specific host. However, we only understand the intricate details and nature of very few of these relationships. By conducting further research on the various cordyceps species and their respective hosts, we can gain a clear understanding of the diverse parasitic strategies and the potential adaptations that may arise.
Next, we are going to touch on the opposite side of the spectrum and focus on a mutualistic relationship between ants and fungus. The leafcutter ants make one or several chambers in their underground nests to store the Lepiotacea fungus, depending on the size of the ant colony. The Lepiotacea fungus acts as the leafcutter colony's primary source of food. The ants farm the fungus by providing it with fresh substrate, soil, and leaf pieces to help it grow. The underground ant colony provides protection for the fungus from other insects that would eat it, as well as pathogens. It has been found that a colony of leafcutter ants rears only one specific clone of the fungus. Worker ants will discriminate between other fungus and their specific clone, and will not change to a different fungus until 1 week after the clone fungus is removed. This specific clone is passed on to new queens when they leave the colony, and this is thought to help stabilize the mutualism by aligning the reproductive interests of the ants and the fungus. All of the fungus-growing ant species manure new mycelium of the fungus with their fecal droplets, which contain fungal enzymes that break down plant material. The ants deposit their fecal droplets onto fresh leaves or directly into the fungus garden, where they are absorbed. One study suggests that obligate manuring of the fungus with the fecal droplets allows the fungus to control the genetic identity of new gardens in the nest, leading to the removal of unrelated fungi and explaining why each colony has only one clone of the fungus.
Understanding species' interactions with each other is important information because it allows us to get a clear understanding of the inner workings of these ecosystems. Taking a look at ants has shown that there are still discoveries being made recently, showing how there is always more to learn about, even in the two interactions we covered. The parasitic interaction we covered is relevant to help highlight the evolutionary arms race that these species compete in to survive, and the various adaptations that follow. The mutualistic interactions help show how species can interact in a way that allows them to work together to have a better chance to survive.
Thank you guys so much for watching. We hope you enjoyed the video and maybe learned something new. If you did, be sure to smash that like button, subscribe, and turn on notifications. We'll see you all next time. Peace. This video was sponsored by Raid Shadow Legends.